Start with a couple of announcements. One is a reminder that problem set 2 was due last night at 9 p.m. I gave an extension to some, but not everybody, so if you haven't handed in your problem set 2, and you do it today, you get docked, there's a late penalty, unless I gave you permission. So it's better late than never. A reminder to everybody that quiz 2 is due tomorrow at 9 p.m. So this is the quiz, you pick it up at the CST desk, you take it in CST, and then you turn it in right there. I think it'll take you between a half an hour and an hour to do. Muscles of the arm, shoulder, forearm, arm, shoulder, and back, and brain structures, neuroanatomy of the brain. That's what's on this quiz. You might be wondering, wait a second, haven't we already been tested on that? And the answer is yes, but repetition is a good way to learn, and so this quiz is a repetition of some things. Not everything, just a repetition of some things. Due 9 p.m. tomorrow, because that's when CST desk closes. And one more announcement, that the Joseph Moore Museum is hosting a series of distanced safe events on Saturday from 10 to 4, 10 in the morning till 4 at night, afternoon. So they've been working very hard on putting together something fun and interesting and sciencey, natural sciencey. So I'm just making that announcement because you might be interested in participating in something fun and sciencey in these days. So that's at the museum Saturday from 10 to 4. All right, so we're gonna do the visual system today, the whole thing. So there's two parts. There's the first part and the second part. So the first part, here are the goals of the first part, then we'll take a break, and then we'll do the second part of the visual system. So we'll start with some neuroanatomy, then some eye anatomy, then some muscle anatomy, then the organization of the retina and how the retina turns light into an electrical signal that we can use. So here is a inferior view of the brain. The eyes are not anatomically correct and they're kind of disturbing, but that's the front. Anterior, ventral surface, posterior, dorsal surface. Left, right. So there's the eyes, which we'll talk about in a minute. The optic nerves are cut, but they were here. So those are the optic nerves. These are the olfactory nerves. Optic nerves from the eye to the optic chiasm, optic tracts from the optic chiasm to a nucleus in the thalamus. This is going to be important now. A nucleus in the thalamus that has a name called the lateral geniculate nucleus. That is a section of the thalamus that processes visual information from the retina. So the retina, optic nerve, optic chiasm, thalamus, specifically the lateral geniculate nucleus of the thalamus. Then there's going to be nerves called the optic radiations from the thalamus to the primary visual cortex in the posterior occipital lobe. So that's an overview of the neuroanatomy that we need. Let's work on the anatomy of the eye. So the clear structure in the anterior surface of your eye is called the cornea. Behind the cornea is a chamber called the anterior chamber, and it's filled with a fluid that is not, for some reason, labeled on this slide. So you need to annotate your slide, because in here is a fluid called the aqueous humor. Aqueous humor. I don't know why that label was left off of this slide, but it was. So light, and it's clear fluid, so light goes through the clear cornea, goes through the clear aqueous humor, goes through the pupil. Again, well, there's something else that wasn't labeled. So label this hole as the pupil. Some of you have this in middle school. The pupil is formed by the iris. So the iris is a circular muscle. It's a sphincter muscle. And so the iris can contract and make the pupil small. You know that that happens in bright light. Or the pupil can dilate. The muscle can relax. The pupil dilates in dim light. That opening is called the pupil. The color of the eye, the thing that opens and closes, is the iris. Brown, blue, hazel, depending on your eye color. Behind the iris is the lens, clear material, and the lens is suspended in the proper position by structures called zonular fibers. Sometimes they're called suspensory ligaments, little tiny fibers that hold the lens in place. And they hold the lens, they are attached to the lens, and they are also attached to this muscle, which is another sphincter muscle. It's called the ciliary muscle. And it's distinct from the iris. It's very close, and it is a sphincter, but they are not the same. The iris controls the pupil diameter, and the ciliary muscle controls the tension on the lens. And you're going to see why that's important in just a few minutes. For focusing the light on the retina. Okay, the light goes through the lens. Physicists, if you took physics and you did any optics, you know what happens with a lens. The image gets inverted, etc. We're not going to focus on physics. The lens, the light goes through the clear lens, and then through the posterior chamber, for some reason not labeled here, posterior chamber. So there's the anterior chamber filled with aqueous humor, and the posterior chamber labeled with vitreous humor. Vitreous means glass-like, and you'll see in the lab when we do eyes on Thursday and Friday that the vitreous humor is like a gel, and the aqueous humor is a liquid. So the name makes sense. Vitreous, it's not glassy, but it is very gelatinous. But it's clear. So the light goes through, and if the lens is doing its job, the image is focused on the retina. The retina is supported by two structures behind it. So there's no support of the retina in front of it. There is support of the retina behind it. The pigment epithelium is right behind the retina, and the choroid is right behind that. And those provide nutrients, vitamin A for retinal. You'll see in a minute why we need, so blood flow and regeneration of the things that happen during vision are the responsibility of the choroid and the pigment epithelium directly behind the retina. So the retina is where we're going to be focusing, because that's what converts light into electrical signals. But it depends on support, biochemical support and physical support from the choroid, from the pigment epithelia in direct contact with it, and the choroid behind that. And behind that is the structure called the sclera, which is the white of the eye. It's a cartilaginous material that gives the eye its structure, circular structure. So that structure, the white of the eye, is called the sclera. the retina has some specific spots, anatomical locations that I do want you to know. There is the fovea. The fovea is a specific part of the retina that is specialized for high acuity vision. When we are focusing our eyes, when we are focusing our vision and our attention on something, like right now, I am focusing my attention on the lens of that of the video camera, looking right at it. I'm adjusting the shape of my lens, and I'm putting the image on my fovea of both eyes. That's where I have my best vision. Now, even though I'm focusing on the video camera, I can see peripherally. I can see that Hannah's over here, and I remember that Logan is over there somewhere, and I can't really see Logan, but I know he's there. And I can see, even though I'm not focusing on Sarah, I can see the color of her shirt. Right? You know, I know she's there, but I'm not seeing her very clearly because she is not on my fovea. Her image is not on my fovea. Her image is in my peripheral vision. So there's lots of peripheral vision, but the fovea is where we see the best. The other spot on the retina is called the blind spot. The blind spot is where the axons from neurons of the retina are going to go out the back. And this is so the blind spot is where the optic nerve, where the axons of cells from the retina go out and form the optic nerve. There's a spot here where we can't see. Now, you could say, well, that's weird. You mean, right now, when I'm focusing on this lens, there's a blind spot. Why don't I see it? Why don't I see two spots somewhere out here where I can't see at all? And the answer is our brain fills it in. All right, let's talk about the musculature of the eye. We're good at controlling the position of our eyes, and we're good at that because we've got a lot of muscles that are very finely controlled for the movement of our eyeballs. So this is a superior view. So here's the nose, I guess, in cross-section. Left eye, right eye. Here's the optic nerve, optic chiasm. So yellow is showing nerve. Brown-orange is showing bone. And red-orange is showing muscle. So let's talk about the muscles and what they do. We have 4 rectus muscles. Rectus just means straight. 4 straight rectus muscles. We've got a lateral rectus, so each eye has a lateral rectus on each side. And when the lateral rectus is contracting, the eyes are moving laterally. I can't do it. I can't make both my eyes go outward at the same time. So I'm just pretending I can, right? Chameleons can do this, but humans can't do this. I can't make my 2 eyes go outward at the same time. But that, if I were able to contract both lateral rectus muscles, that's what would happen. There are medial rectus muscles. Medial rectus muscles on both eyes. And now you can get the idea. If I contract both my medial rectus muscles, I would go cross-eyed. And I can do that, but I don't want to. So lateral rectus muscles moves the eye outward, and medial rectus muscles move the eye inward. And now you can see that these 2 muscles working together allow me to move my eyes to the right or to the left. If I want to move my eyes to the left, I am contracting the lateral rectus of my left eye, and I'm contracting the medial rectus of my right eye in order to move my eyes this way. To move my eyes to the right, I'm contracting the lateral rectus of my right eye and the medial rectus of my left eye in order to move this eye towards the midline and move this eye away from the midline. Okay, those two of the rectus muscles. The other two rectus muscles are the superior rectus muscles, superior rectus, and inferior rectus. So the superior rectus has been cut away on this side so that we can see the inferior rectus. You can sort of see it here, the superior rectus attached to the top of the eye, and the inferior rectus attached to the bottom of the eye. And now you can imagine what these do. The superior rectus muscles, if they contract, move my eyeballs up. And so elevation of my eyes. And contraction of the inferior rectus, move my eyes down. Depression of my eyes. So those are the four most important muscles of the eye. We also have two more, so there's 6 muscles in total for each eye. The 4 rectus muscles, lateral, medial, superior, and inferior. And now there are 2 obliques. There's a superior oblique and an inferior oblique. Inferior oblique is kind of hard to see. Inferior oblique, right there. So the inferior oblique attaches out here and up here. And the superior oblique attaches here, goes through this structure called the trochlea, and then attaches down here. What do these do? These are important for rotating the eye because we want to keep our image level. And they support. So the superior oblique assists in moving the eye laterally as well as rotationally. The physics of it get strange. We don't think of our eyes rotating very much, so they're less important. I want you to know the functions, I want you to know that they exist, and I want you to know that their functions is rotation and tweaking of lateral and superior, lateral movement, medial movement, superior and inferior movement. They tweak that. But the 4 rectus muscles do most of the heavy lifting. It's not very heavy, it's most of the work in moving the eyes. Let's talk for a minute, questions about the musculature of the eyes. This is anatomy, but this is not on quiz 2. This will be on quiz 3. Okay, let's talk about innervation. Let's talk about the innervation of the eye. So here is a sagittal view. So whenever you're looking at the eye, you have to know, am I looking at it? Am I looking at the eye in a horizontal section? Or am I looking at the eye in a sagittal section? Or I suppose there could be a coronal section. You just have to know your orientation. So this is a sagittal view of the eye. Let's just say it's the left eye being looked at from the right. It's very complicated. Let me make it make sense to you. The most important nerve that innervates the muscles of the eye. Remember, the optic nerve is important for taking sensory information from the eye to the brain. But now we have to think about the nerves that innervate the muscles, the motor nerves. The most important nerve for controlling the movement of the eye is the oculomotor nerve. So highlight or circle this name right there. The oculomotor nerve, that's cranial nerve 3. Old Oprah's occupation. Oculomotor nerve. The oculomotor nerve innervates, so let's just follow Pink. It innervates the superior rectus. It innervates the inferior rectus. It innervates the inferior oblique. It innervates the medial rectus. That's not shown here, but it does. It innervates the medial rectus. It also innervates muscles that control the eyelid well, let's just say the eyelid, right? I'm not going to test you on that, but I could test you on knowing that the oculomotor nerve innervates the medial rectus, the inferior rectus, the superior rectus, and the inferior oblique. 4 of the 6 muscles. The other muscles, so the superior rectus, the superior oblique is innervated by the trochlear nerve. So now, if you know the name trochlear, you remember that strange little structure, the trochlea. So the superior oblique goes through the trochlea and is innervated by the trochlear nerve. The lateral rectus, which is not shown here, is innervated by the abducens nerve. So remember, moving the eyes outward is abduction of the eye. Moving the eyes outward, abduction, triggered by the abducens nerve contracting the lateral rectus muscles. So the abducens nerve controls the lateral rectus muscles, the trochlear nerve controls the superior oblique muscles, and the oculomotor nerve controls all the others. It also indirectly, so watch this, it also indirectly controls the contraction of the iris and the ciliary muscle. So the oculomotor nerve, through this secondary connection, controls the diameter of the pupil and the focus of the lens. You don't need to worry about the arteries. You don't need to worry about the sympathetic plexus. You don't need to worry about the trigeminal ganglion here. But you don't have to worry about the ciliary nerves. But I do want you to know oculomotor nerve and what it innervates, trochlear nerve and what it innervates, abducens nerve and what it innervates, and what those muscles do. Questions about innervation? Let's talk about focus. So, right now, I'm focusing on my fingertip. I can make my eyes aren't the best, and I'm wearing bifocals, etc., so I need assistance. But I can focus on my finger. What is it? Maybe 8 inches? Very difficult for me to focus any closer than that, so about 8 inches. I can also focus on the lens of the camera. I can focus on Carpenter, way over there. So I can make a nice sharp image of Carpenter. That's pretty easy. And I can make a pretty sharp image of my finger right here. So I can focus a pretty long distance, pretty wide distance range. How do we do that? So let's talk about a distant image. I'm focusing on that house on the other side of National Road over there. Very far away. The light is a long distance. In order to focus that light on my retina, I need my lens to be flat. If my lens is too round, it's going to focus it in the wrong place. So I need my lens to be flat. The way I get my lens to be flat, so my lens is flat, that is to say, the zonular fibers that surround my lens are pulling my lens flat. My lens will naturally rebound into sort of a round state, roundish state. So the lens wants to be round, so to speak, but the zonular fibers put tension on it and pull it flat. Contraction of the ciliary body, ciliary muscles. Scratch that. Relaxation of the ciliary body puts tension on the fibers and makes the lens flat. So when I'm focusing on a distance object, I'm relaxing my ciliary body and putting tension on the fibers, pulling the lens flat. When I need to focus on a nearby object, I need a round lens. I need to contract the ciliary body in order to close the sphincter. That puts less tension on the zonular fibers. So, contraction of the ciliary sphincter, less tension on the fibers, allows the lens to round up, and I can focus on a nearby object. And if it's not right, if I'm not focusing right, if I'm using distance focus to try to see my finger here, it's out of focus. You know, I'm intentionally focusing on the house on the other side of National Road, but I'm putting my finger in the field of view. I can see it, but it's not in focus. It's not in focus because the light is not focused on my retina. We need to focus this image on the retina in order to see it clearly. In fact, in order to see it really clearly, you have to focus the image not only on the retina, but on the fovea of the retina. So I do want you to know that focus on a distance object requires relaxation of the ciliary muscle, tension on the lens, and a flat lens. And focus on a nearby object requires contraction of the ciliary muscle, slack tension, low tension on the zonular fibers, and a rounded lens. The nerve that controls this is the oculomotor nerve, and the secondary nerves from the ciliary ganglia. So, oculomotor nerve, contracted ciliary body, slack tension, round lens, focus for a near object. If I focus on a near object, so I'm focusing on my finger, and I can see Meg behind my finger, but she's not in focus because her image is not focused correctly. Now I can look at Meg and see her in perfect focus. Questions about focus? All right, so now we are going to get into the most interesting part of this, I think, and that is how we turn light into vision. How we turn light into electrical signals that our brain can interpret, right? The light's not getting into my brain. I'm not shining a picture, a camera, or a projector somewhere in my brain. My brain is interpreting electrical signals, which comes from this image that I'm forming of you all. How do we do that? Okay, so this is part of the story. How we turn light into vision. Let's get oriented. This is the back of the eye. This is the front of the eye. Because here's the choroid, and here's the pigment epithelium. What's behind the choroid here that is not labeled would be the sclera. So sclera, choroid, pigment epithelium, retina. The thickness of the retina is exaggerated compared to the thickness of these two numbers. Retina. So light came through the cornea, the aqueous humor in the anterior compartment, the lens went through the pupil, went through the lens, went through the vitreous humor in the posterior chamber, and now is coming in this way. So the first cells that the light hits is not the cell that does the phototransduction. You're going to see this in the middle. It's the last cell that the light hits that turns it into an electrochemical signal. So the light actually goes through some transparent layers of the retina and then gets transduced into an electrical signal by the photoreceptor cells. So these are the photoreceptor cells, rods and cones. Rods and cones. This is a cartoon, of course. This is an actual electron micrograph, rods and cones. So these cartoons come from something reality-based. The rods and the cones are going to transduce photons, light, into electrochemical signals. And then it's going to get processed by several cell types in the retina. So the image gets formed, and the image gets transduced into electrochemical signals in the retina, and then it's immediately, right there in the retina, it's going to get processed before it even gets to the brain. So most neuroanatomists consider the retina part of the brain. So let's talk, so here are the cell types. We've got rods and cones, the photoreceptor cells. The next cell in the pathway are called bipolar cells. The next cell in the pathway are called the ganglion cells, and these are the cells that have axons that make the optic nerve. The rods and the cones do not have axons. The bipolar cells do not have axons. The ganglion cells have the axons. So now, using your knowledge from all the way from last week, are rods and cones long receptor cells or short receptor cells? Short. They're short receptor cells. These cells do not have an axon. They transduce light and they release chemicals, neurotransmitters, at the synapse, but they don't fire action potentials. Bipolar cells are also short. They're not even receptor cells, and they are neurons, but they don't have an axon. This is not really an axon. It's not until we get to the ganglion cells that we have a neuron with an axon that forms the optic nerve. So the through pathway is photoreceptor, either rod or cone, bipolar, and we're going to see two different types of bipolar cells, and then ganglion cells. At each of those connections, there is a complicated set of synapses. So here's the synapses between this cone and this bipolar cell. And here are the synapses between this bipolar cell and this ganglion cell. So a synaptic layer and a synaptic layer, where lots of processing is going to go on. There are cells, in addition to this through path, there is a path for lateral inhibition. And in the retina, lateral inhibition is done by two types of interneurons called horizontal cells and amacrine cells. I do want you to know the names of the cells that form lateral inhibition in the retina. Horizontal cells do lateral inhibition at the first synapse, and amacrine cells do lateral inhibition at the second synapse. In general, what does lateral inhibition do? What is the function of lateral inhibition, speaking broadly about sensory physiology? Somebody speaking. Anybody? What does lateral inhibition in the somatosensory system do? It sharpens our sensation. It reduces receptive field size. Sharpens our sensory abilities. And it's the same thing here. We can form really sharp images, in part because we have extensive lateral inhibition in the retina. Lateral inhibition sharpens the receptive fields in the retina. Here's an electron micrograph that shows that cartoon is not making it up. So here's the sclera, here's the choroid, here's the pigment epithelium. Actually, it shows that this cartoon is actually pretty close. The thickness of the choroid and the pigment epithelium relative to the retina. Here's the pigment epithelium, and the rest of this is the retina. So it's not so far off. Here are the rods and cones. So this is this layer here. Here are the cell bodies of the rods and cones. So this layer and this layer are the rods and cones. Really, this layer and this layer. So these are the rods and cones, the outer segments and the cell bodies. Plexiform is where the synapses are. So this layer is this layer. Inner nuclear layer, cell bodies, cell bodies are all these cell bodies. The cell bodies of the bipolar cells, the cell bodies of the horizontal cells, and the cell bodies of the amacrine cells are all in that other cell body layer. I'm glad you haven't been writing this down. I am not going to ask you to memorize the names of these layers. I'm not going to do that. So you do not have to memorize plexiform layer or nuclear layer. I'm just showing you that it's real. Synaptic layer, and then here are the ganglion cells that have the axons that form the optic nerve. Let's talk for a minute about the fovea. So what are we seeing here? We're talking about the number of receptor cells, not receptor molecules, but receptor cells per square millimeter of retina. So a little area, a square millimeter can have 160,000 photoreceptor cells in it. And this is as distance really, angle from the fovea. So we start at the fovea. Let's look at the fovea. At the fovea, we have 150,000 receptor cells per millimeter, but they are all cones. Our cones are the fovea is where most of our cones are expressed. Outside of the fovea, even just a few degrees, just a few microns away from the fovea. So we're moving away from the fovea, and that's where we have a large number of rods. The cones are specialized for color and for high acuity vision, very sharp vision. So we've specialized our fovea in order to see color and in order to see sharp. Outside of the fovea, where the rods are, the rods do not see color. They're not as sharp. They don't form as sharp an image. But they respond very quickly. And they respond very quickly to movement. Moving an object in my peripheral vision gets my attention very fast because the rods are fast. So our best vision is when we focus an image on our fovea. But cones are specialized for color, but they have a downside. They require relatively bright light. Rods don't see color, but they see dim light really well. So if you're trying to navigate in the dark, your peripheral vision is better than your fovea vision. Some of you might have had this experience if you're trying to look at stars at night. You know, the night sky is pretty dark, right? And you're looking at those spots of light. And a bright spot of light, you know, if you're focusing on a bright spot of light, a star, you can see it. But there are some stars that are quite dim. And if you try to focus on a dim star, it disappears. But if you avert your gaze just a little bit away and put the image of that star in your peripheral vision, you can see it. Because your peripheral vision is better for dim objects, like dim stars. So if you talk to somebody in the physics department who teaches astronomy, like Seth, he might tell you, yeah, astronomers develop pretty good peripheral vision in order to see objects that are dim, not where we're focusing, but dim sort of outside of our main focus. Anyway, the main point was just interesting. The main point is cones in the fovea for color vision and high acuity vision, rods outside of the fovea for dim light vision and motion vision. All right, here is an image of a rod and a cone. The working end, where light is transduced into an electrical signal, is in this outer segment. So the inner segment is where the cell body of the synapse is. The outer segment is where the photoreceptor molecules are of these photoreceptor cells. The photoreceptor molecule is a GPCR. So here is a cartoon of a G protein coupled receptor embedded in the membrane. Here is the G protein that is associated with this GPCR with its lipid linking. So here is the alpha subunit, and here's the beta gamma subunit. These receptors and G proteins have specific names. The receptors are called opsins. So there's rhodopsin, which is expressed in rods, and then there's going to be 3 other opsins expressed in cones for giving us color vision. So the receptors are called opsins, rhodopsin, and then red, green, and blue opsin in the cones. And then the G protein has a name. It's called transducin. So the name of the G protein in the visual system is transducin. So how does this receptor work? There is a molecule covalently linked to the opsin called retinal. retinal. It looks like retinal, but it's retinal. So embedded in the opsin is retinal. Retinal is a light detector molecule. When light hits it, it undergoes an isomerization from cis to trans. You don't have to know that. Retinal is what undergoes a conformational change in response to a photon hitting it. Conformational change of the retinal, conformational change of the opsin, conformational change of the G protein, and now we're off and running with the same G protein story that I'm going to elaborate in a minute. But you already know where I'm going to go. GDP dissociation, GTP association. You don't know where it's going to go yet, but you already know this story. Retinal and opsin are what turn light into something that we receive, a receptor response. We see color because the opsins, we've made mutations in opsins in order to change the spectral sensitivity of the opsin. So, for example, rhodopsin, the opsin in our cones, in our rods, the opsin in our rods is called rhodopsin, and it has a spectral sensitivity, and it detects blue-green light best. It can detect yellow light, but poorly. It can detect purple light, but poorly. It's best at detecting blue light. In our cones, so each one of our cones expresses a certain opsin, not rhodopsin, but another opsin, and there are specific amino acid differences between the opsins and rhodopsin. Give the retinal in those opsins a different spectral sensitivity. So all of them have the same retinal, they have the same molecule, but because the retinal is embedded in an opsin, and because there are amino acid changes in the opsin that change the interaction of the retinal with the opsin, the so-called blue cones, of course they're not blue in color, but they have evolved the highest sensitivity to blue-purple light. And the green cones are not green, but they have amino acid specificity that gives the retinal in that opsin the highest specificity to green light. And we have red cones that express a red opsin that tweaks the retinal. So its highest sensitivity is actually in the yellow spectrum, but we call these red cones because they're the only ones that really see red very well. You can see out here in the red, looking at a red rose. Let's see somebody wearing anything bright red. Eric's water container is red-orange. It's out here somewhere. And I can see it because I've got cones that are specialized for seeing that color. I'm looking at Sarah's green shirt. So that green is somewhere around here. I'm not using my rhodopsin very much. I'm certainly not using my blue cones. I'm using my green cones and my red cones to form an image that my brain interprets as light green. Let's see if there's any other primary colors in the Andrew's pink shirt. Pink is a light red, so I'm using my red cones to see Andrew's pink shirt. Different cones express different opsins with specific mutations, with specific single nucleotide polymorphisms, single amino acid changes that render the retinal sensitive to specific colors of light with a spectrum, with a range. So even though red is really best detects yellow light, it also detects red light. Now what I'm going to do is get into the mechanism by which receptor cells convert light into an electrical signal. That's where we're going. So we're going to focus on a cone. Cones have this sort of cone-like structure of membrane. The folding of whenever you see folding of membrane in any physiological system, it's generally to give more surface area in order to express more something. And in this case, more surface area is expressing more receptors, more receptor molecules. So I have to be careful. This is a receptor cell, and now we're talking about receptor molecules. Okay? Here is the opsin. That's the receptor molecule in this tooth of this cone. Here's the opsin. There's the retinal in its cis configuration. Okay, before I get into what light does, I need, in this case, to talk about what happens in the dark. So in the dark, there is an enzyme called guanylyl cyclase that is constantly making cyclic GMP from GTP. In the dark, the cells are metabolically active, converting GTP into cyclic GMP. The cyclic GMP opens a nucleotide-gated cation channel. In the dark, the production of cyclic GMP opens a cyclic nucleotide-gated ion channel that is selective for sodium and calcium. In the dark, there is a high permeability of these channels because they're open by the high amount of cyclic GMP, and there's high sodium influx. So in the dark, do these cells have a highly depolarized, quote, resting potential, or are they hyperpolarized relative to, say, normal cells? But you already know the answer to this. You can apply from exam 1, your knowledge. Are these cells in the dark depolarized compared to a typical cell or hyperpolarized compared to a typical cell? Depolarized, because there's relatively high amount of sodium and calcium influx. That makes these cells relatively depolarized in the dark. And like other cells, depolarized cells are releasing a lot of neurotransmitter. We're going to get to that in a minute. Now, let there be light. Light hits the retinal, conformational change of the opsin, conformational change of the transducin. What's not shown here? GDP dissociating and GTP associating. Alpha separating from beta gamma. The active alpha with the GTP bound activates an enzyme. So here we are. Light, conformational change, GDP dissociation, GTP association. alpha activation activates an enzyme called phosphodiesterase. Okay, you've seen this before. Phosphodiesterase, what does it do? It cuts cyclic GMP into just ordinary GMP. This enzyme reduces the concentration of cyclic GMP. That's how our opsins work. Light, story, enzyme cuts cyclic G. So now if cyclic G concentrations go down, what happens to the amount of channel opening here? It goes down. What happens to the membrane potential? If we close these channels because we've broken up we've reduced cyclic G. What happens to the membrane potential? Does it go more depolarized or does it go more hyperpolarized? Hyperpolarized. Light. So here's the cell in the dark. A relatively depolarized membrane potential, right? You know normal is minus 60 or minus 70. Here it is at minus 35. A depolarized dark membrane potential. Light causes a hyperpolarization. What do you suppose the difference is between this what's the difference in the stimulus that produced this hyperpolarization or this hyperpolarization or this hyperpolarization? What's the difference in the stimulus? Brightness, right? This is a graded potential. A bright light produces a large hyperpolarization. A dim light produces a small hyperpolarization. So in the dark, there is a lot of neurotransmitter release. Let's go back to the synapse. In the dark, there is a lot of neurotransmitter release from this synapse. It's happening in the dark, even while you're sleeping. It's dark. Your eyes are closed. It's dark. This synapse is releasing neurotransmitter. Your photoreceptor cells are having to make a lot of GTP in order to make a lot of cyclic GMP. When light hits it, hyperpolarization reduces the neurotransmitter release. What channel is important here for neurotransmitter release? I didn't say it, but you already know it. Voltage-gated calcium channels. If you depolarize this cell, calcium influx here, release of neurotransmitter. Light, receptor, conformational change, phosphodiesterase, cutting cyclic G, closing the channels, hyperpolarizes, closes the voltage-gated calcium channels, less neurotransmitter release. That's the first step of phototransduction. The first chemical step from light, lots of receptor information that I do want you to know, hyperpolarization, closing voltage-gated calcium channels, reduction in neurotransmitter release. Light is turning into a electrochemical change. Let's stop here. There are some clickers that I'm going to let you do as homework. Okay, so let's take a good 10-minute break here. Form your knowledge. Maybe work on the clickers. And then we'll come back and turn that electrochemical signal of the photoreceptor cell into vision in the next